Right now, as you're listening to my voice, there is literally a microscopic electrical storm just raging inside your head.
Oh, absolutely.
I mean, every word I'm saying, every concept you understand, and, even the visual image you might be forming right now of your own brain, all of that is being powered by billions of these tiny biological sparks.
It's wild to think about.
Right. It's the ultimate meta experience. Your brain is literally using its own physical hardware to understand a conversation about that exact same hardware.
Yeah, it really is staggering when you stop to, actually think about it, because we spend so much time talking about psychology in the abstract, like our emotions, our behaviors, passing thoughts. But underneath all of that software, there is very real, very physical, biological machinery at work.
And that is exactly our mission for today's deep dive.
Yeah.
We are zooming all the way in, like microscopically in, to look at the literal cells of the nervous system.
I'm ready.
So, to playfully set the dynamic for you, the listener, today I am officially declaring myself the eager student, and, well, you are our lecturer.
I accept the role.
Perfect. And our syllabus today is drawn directly from Section 3.2 of Chapter 3 from Psychology 2E. So, lecturer, where do we even begin our inventory of this microscopic world?
Well, we have to start by meeting the cast of characters, right? If we're looking at the nervous system, there are essentially two basic types of cells we need to care about.
Okay.
You've got your glial cells, often just called glia, and your neurons. Now, for the longest time, the scientific community had this very specific, almost dismissive view of glia.
Really? Dismissive how?
Well, they were just seen as the support staff. They provide the physical scaffolding that holds the nervous system together.
Ah, I see.
Yeah, they help neurons line up. They provide insulation, transport nutrients, clean up waste, that sort of thing.
So they're doing all the grueling heavy lifting to keep the environment stable, but they aren't the ones actually, processing our thoughts.
Precisely. That's where the neurons come in. They are the interconnected information processors.
It's ours of the show.
Exactly. They are the essential units that handle all the actual tasks of the nervous system. And you have about 100 billion of these neurons at birth.
Wow. 100 billion.
Yeah, but what's absolutely fascinating here is how our understanding of the ratio between these two cells has, well, it's recently been turned entirely upside down.
Wait, how so?
So for decades, textbooks claimed as an absolute fact that glial cells massively outnumbered neurons.
Like by how much?
Usually citing a 10-to-1 ratio. But more recently, groundbreaking research from Suzanna Herculano-Houzel's laboratory completely shattered that assumption.
Oh, wow.
Yeah, she provided vital evidence that the ratio in the human brain is actually nearly 1-to-1.
Okay, wait, let me make sure I'm wrapping my head around this.
Right.
If there's a 1-to-1 ratio, that means for every single neuron firing off an electrical signal, there is a dedicated glial cell supporting it.
Exactly 1-to-1. Yeah.
So to use an analogy, if the neurons are the rock stars playing the actual music, the glial cells are the roadies who set up the stage, tune the guitars, and, pack up the gear afterward.
That is a brilliant way to look at it, actually. The rock stars get all the glory, obviously, but the show physically cannot happen without an equal number of roadies working behind the scenes.
But pushing back on this just a bit as a student, how did the entire scientific community get that ratio so spectacularly wrong for so many years?
It's a great question.
I mean, why did everyone just blindly accept the 10-to-1 myth?
Well, it's a perfect example of how science is bound by the limitations of its tools, and the danger of untested assumptions.
Right.
Early counting methods were incredibly flawed. Scientists would take a tiny slice of the brain where glial cells happen to be highly concentrated, count that one small sample, and then broadly extrapolate that ratio to the entire brain.
Oh, so they just assumed the whole brain looked like that one slice.
Exactly. So Herculano-Houzel had to invent a completely new method. She essentially dissolved brain tissue into a uniform soup.
Brain soup.
Literally, yeah. A soup where every single cell nucleus was evenly suspended, and only then could they be accurately counted.
That is incredible. And I imagine that new 1-to-1 ratio changes how we view ourselves biologically, right?
It really does. It proves that human brains are far more similar to other primate brains than we previously thought.
Oh, interesting.
Yeah, we used to think we had this magically unique glia-heavy brain that made us smarter. It turns out we don't. We just have a very large standard-issue primate brain.
Okay, so if these neurons are 100 billion rock stars, and they are doing the actual heavy lifting of processing every thought and sensation you and I have,
Yeah.
how are they physically built to handle that much data?
They're very unique.
Right, because they can't just be standard round microscopic cells.
No, not at all. They are highly specialized. A neuron's anatomy is completely built around the intake and output of information. Let's map it out from the outside in.
Oh, good on me.
The outer surface is a semi-permeable membrane. And this membrane is incredibly picky.
Picky how?
It allows smaller molecules, or molecules without an electrical charge, to pass through effortlessly, but it actively stops larger or highly charged molecules.
So it's less of a wall and more of a highly selective bouncer at a club, deciding exactly who gets VIP access.
Exactly. Perfect analogy. Now, inside that membrane is the soma, or the cell body, which holds the nucleus.
Got it.
But a neuron doesn't just sit there in isolation, right? It has to talk to its neighbors. So, branching out from the soma are these sprawling root-like extensions called dendrites.
Dendrites, okay.
If the neuron is a processor, the dendrites are the input sites. This is where it receives incoming signals from other neurons.
Okay, so input comes into the dendrites, moves to the soma, and then how does it send the message out?
The signal is transmitted electrically away from the soma down a single major extension called the axon.
Okay.
And this is where the engineering gets wild. Axons can be microscopic, just a fraction of an inch, or they can be incredibly long.
How long is incredibly long?
You have single axons stretching all the way from the base of your spine down to your big toe.
Wait, a single cell extension is several feet long?
Yes,
Yeah.
exactly.
That sounds like a massive logistical nightmare. If you have an electrical signal trying to travel down a microscopic biological wire that spans an entire human leg, wouldn't this signal just degrade or short out before it gets to the end?
It absolutely would, which is why evolution provided a solution. And this brings our roadies, the glial cells, back to center stage.
They're back.
Right. In many axons, glial cells actually wrap themselves around the axon to form a fatty substance called the myelin sheath.
The myelin sheath.
Yes. This sheath is an electrical insulator. By coating the axon, it drastically increases the speed and efficiency at which the electrical signal travels.
Okay, makes sense.
But it isn't one solid tube. There are microscopic, evenly spaced gaps in the myelin sheath all the way down the axon, known as the nodes of Ranvier.
I see. So the myelin sheath acts exactly like the rubber coating on a phone charger cable.
Exactly like that.
It keeps the electricity flowing quickly and safely right where it's supposed to go.
Precisely. And Section 3.2 highlights just how critical this insulation is by looking at what happens when it breaks down.
Because it doesn't always work perfectly, right?
Right. If that myelin sheath frays or is destroyed, the consequences for human health are devastating. The textbook mentions a genetic disorder called PKU, which causes a reduction in myelin.
Oh, wow.
Yeah, without proper insulation, information can't travel effectively, leading to severe cognitive deficits, exaggerated reflexes, and seizures.
It's terrifying to think that a microscopic loss of fat around a single cell can completely alter someone's cognition.
It really is.
The textbook also mentioned multiple sclerosis, right?
Yes. MS is a tragic and stark example. It's an autoimmune disorder where the body's own immune system mistakenly attacks the myelin sheath.
So it's actively destroying its own insulation.
Exactly, causing large-scale loss of insulation throughout the nervous system. The electrical signals experience massive interference.
It's awful.
For you listening, if you've ever known someone with MS, you know this microscopic breakdown leads to very macroscopic symptoms.
Like what?
Dizziness, profound fatigue, loss of motor control, sensory dysfunction. It is a stark reminder of how dependent our lives are on these microscopic fatty coatings.
That really grounds it. It's the little difference between walking smoothly
Yeah,
and losing control of your body.
absolutely.
So we've mapped out the physical wiring and the insulation, but what about the spark itself? Like, what is the actual electricity flowing through these biological cables? How does a neuron actually fire?
Oh, this is a beautiful piece of biological engineering. And to understand it, we have to talk about tension.
Tension, okay.
The neuron exists in a fluid environment. There is fluid inside the cell, the cytoplasm, and extracellular fluid outside it.
Right.
That semi-permeable membrane we mentioned earlier, it keeps these two fluids separate. And that separation is critical because the electrical signal entirely depends on these two fluids being electrically different.
Right. So they have different charges, which creates potential energy.
Exactly. This difference in charge across the membrane is called the membrane potential. Now, the electrical charge is created by ions, which are charged molecules dissolved in the fluid.
Okay, I'm following.
When a neuron is just resting, waiting for a signal, it's in a state called the resting potential.
I love the analogy Section 3.2 uses here. It compares the resting potential to a rubber band stretched tight, just waiting to snap.
It's a perfect analogy because it requires active energy to maintain that tension. At the resting potential, you have a massive concentration of sodium ions, which are positively charged, waiting outside the cell.
And they want in.
Oh, they are highly concentrated, and they really want to get in. Inside the cell, you have potassium ions, which are also positively charged, and they want to get out.
Right.
But here is the trick. The inside of the cell is kept slightly negatively charged compared to the outside.
Wait, if I'm thinking about this logically, how can the inside stay negative if it's full of positive potassium ions?
Because of an incredible mechanism called the sodium-potassium pump.
Oh, the pump. Okay.
It's working constantly, burning energy, pushing 3 positive sodium ions out of the cell for every 2 positive potassium ions it lets in.
Oh, I see.
Because it's constantly kicking out more positive charge than it takes in, the net charge inside stays negative.
Got it.
So picture the scenario: you have a high concentration of positive sodium outside. It's looking at the negatively charged interior of the cell, and the laws of physics are screaming at it to rush inside. Both the concentration gradient and the electrical gradient are pulling it inward.
It's a stretched rubber band.
Yeah.
The tension is built. So what snaps it?
A signal arrives at the dendrites from an adjacent neuron. If that incoming signal is strong enough, it pushes the internal charge of the cell up to a very specific tipping point called the threshold of excitation.
The tipping point.
The millisecond that threshold is hit, the microscopic gates on the cell membrane blast open. All that positive sodium violently rushes into the cell.
Whoa.
The internal charge spikes from negative to heavily positive. That massive positive spike is the action potential.
The spark.
The spark. It peaks rapidly. The sodium gates snap shut, the potassium gates open to let positive charge out, and the cell quickly repolarizes.
It goes back to normal.
Well, it actually overshoots and hyperpolarizes, becoming extra negative for a split second before the sodium-potassium pump settles everything back to the resting potential.
That's a lot of action for a split second.
And this entire positive spike, the action potential, races down the axon toward the terminal buttons.
And because of those gaps, the nodes of Ranvier, the signal doesn't just seep down the line, it physically leaps from node to node, supercharging the speed.
Exactly.
The textbook mentions that this action potential is an all-or-none phenomenon, exactly like sending a text message.
I love that comparison.
Right, you can't send a text. You can type it out, you can hover your finger over the button, but until you hit send, nothing happens. But the instant you hit send, the message is gone at full strength, and you absolutely cannot pull it back.
That binary nature is crucial. The neuron either fires fully or it does not fire at all. There is no intermediate state.
But this raises a huge question for me. If every single signal is all or none, and every single signal fires at the exact same full strength down the axon, how does my brain know the difference between a feather lightly brushing my arm and someone punching me in the shoulder?
That's a really common point of confusion.
If the microscopic spark is identically strong every time, shouldn't they feel exactly the same?
That is the exact paradox early neuroscientists wrestled with. It's true that the strength of an individual action potential never fades. It travels at full power.
Right.
That's why an injury to a distant body part, like a stubbed toe, feels just as sharp and immediate as a scratch on your nose. The signal doesn't weaken over the long journey up your leg.
Makes sense.
But to answer your question, your brain doesn't determine intensity by the strength of a single neuron signal. It determines intensity by the frequency of the firing and the number of neurons firing.
So it's not one voice shouting louder. It's the difference between a single person casually saying a word and a choir of thousands screaming it in rapid-fire succession.
Exactly. A light brush of a feather might cause a few scattered neurons to fire action potentials at a relatively slow rate.
Okay.
A hard punch causes thousands of neurons to fire action potentials incredibly rapidly. Your brain decodes that high volume and high frequency as intense pain.
That makes total sense. Okay, so this all-or-none electrical spike races all the way down the axon to the very end, the terminal buttons. But here's the catching. Neurons don't actually physically touch each other. There is a microscopic void between the end of one neuron and the dendrite of the next. How does the electrical signal cross empty space?
What's fascinating here is that the story shifts dramatically because the electricity doesn't actually cross the gap.
It doesn't?
No. The electrical event literally converts into a chemical one. That microscopic gap is called the synaptic cleft.
Okay, the synaptic cleft.
When the action potential reaches the terminal button, it forces tiny sacs called synaptic vesicles to rupture. These vesicles spill chemical messengers directly into the synaptic cleft.
Then these are our neurotransmitters.
Yes. These neurotransmitter chemicals float across the incredibly small fluid-filled gap and bind to corresponding receptors on the dendrite of the receiving neuron.
So they just float across and grab on.
Right. These receptors are specialized proteins on the cell surface, and they come in very specific shapes.
Which is why the literature always uses the lock and key analogy.
Exactly.
A specific neurotransmitter will only fit a specific receptor, just like a key fits a specific lock. If it fits, it binds. And if that binding creates a strong enough signal, it triggers the threshold of excitation in the new neuron, and the whole electrical storm starts all over again.
Exactly. Once the message is delivered, the chemicals left floating in the synaptic cleft have to be cleared out so the system can reset.
How does it clean up?
Some drift away, some get broken down by enzymes, but a critical process is called reuptake. This is where the neurotransmitter is actively pumped right back into the original neuron that released it, clearing the synapse.
Okay, let me push back on this whole system, though. Because as I look at this biological process, it seems wildly inefficient.
How do you mean?
Why go through all this trouble? I mean, why take lightning, convert it into a chemical puddle, float it across a microscopic moat, fit it into a lock, just to turn it back into lightning on the other side?
It does sound like a lot of extra steps.
Right. Why wouldn't evolution just hardwire us completely with electrical connections? Wouldn't a purely electrical grid be massively faster?
It would be faster, yes. And the textbook actually notes that there are a few rare, entirely electrical synapses in the human body connected via gap junctions purely for sheer unavoidable speed.
Oh, so we do have some.
We do, but if your entire brain were hardwired electrically, it would be an absolute disaster for your survival.
How is that?
Because you would lose all control. Hardwiring means every signal triggers the next, inevitably.
Like a domino effect you can't stop.
Exactly. The chemical gap, the synapse, is what provides essential on and off states. By using reuptake to clear the synapse, the brain can sharply terminate a signal. Ah, okay.
More importantly, the chemical step allows for infinite modulation. The sending neuron can release a flood of neurotransmitters, or just a trickle.
So it has volume control.
Yes. The receiving neuron can sprout more receptors to become more sensitive, or pull them back to become numb.
So the chemical gap is what gives the brain its flexibility.
Exactly. If we were entirely hardwired, we'd be rigid, unchangeable robots reacting automatically to every stimulus.
That sounds terrible.
It would be. The chemical synapse allows for learning. It allows for memories to form by strengthening specific connections. It allows for your mood to shift. The gap is where your humanity actually happens.
Wow, the gap is where humanity happens. But I imagine that incredible flexibility also creates a massive vulnerability.
It certainly does.
If our entire experience of reality relies on this delicate chemical jump across a tiny gap, what happens when the chemicals get out of balance?
That is the crucial question.
Yeah.
And it brings us to how biology directly informs clinical psychology.
Psychologists who take a biological perspective assert that many profound psychological disorders like severe depression or schizophrenia are deeply rooted in physical imbalances within these exact neurotransmitter systems.
So it's a hardware issue manifesting as a software glitch.
That's a perfect way to put it.
So if an imbalance is causing the glitch, I assume this is exactly where psychotropic medications step in. If a patient's brain isn't naturally producing enough of a specific key to open the locks, you'd need a drug that can act as a chemical stand-in,
Right.
a mimic to, pick the lock.
You've just perfectly described an agonist. Psychotropic drugs generally fall into a few categories based on how they interact with that lock and key system at the receptor.
Okay, an agonist.
Yeah, an agonist is a chemical that mimics a neurotransmitter. It's similar enough in shape to the natural neurotransmitter that it can fit into the receptor site and activate it.
So it's stepping in to do the job when the natural chemical is depleted.
Yes. For example, Parkinson's disease is a progressive disorder physically associated with dangerously low levels of the neurotransmitter dopamine.
Right, I've heard that.
So a standard treatment protocol involves dopamine agonists. These drugs circulate in the brain, bind to dopamine receptors, and mimic the missing dopamine, which helps restore the patient's motor function.
Got it. Now, what if the problem is the exact opposite? What if the brain is producing too much of a neurotransmitter and the system is just completely overstimulated?
Then you would prescribe an antagonist. An antagonist does the exact opposite of an agonist.
Okay.
It fits into the log, but it doesn't turn it. It just sits there, physically blocking the receptor site, preventing the actual overabundant neurotransmitter from getting in and activating the neuron.
That makes sense.
Certain symptoms of schizophrenia, for instance, are associated with overactive dopamine neurotransmission. So the antipsychotic medications used to treat those symptoms are often dopamine antagonists. They block the receptors to quiet the system down.
Okay, so agonists mimic to boost the signal, antagonists block to dampen the signal. But the textbook mentions a third biological hack that works entirely differently: reuptake inhibitors.
Right. Remember how we said the chemical synapse is normally cleared out by reuptake?
Yeah, pumping the neurotransmitters back into the cell they came from?
Exactly. Well, a reuptake inhibitor violently interrupts that cleanup process. It blocks the transportation back into the sending neuron.
Meaning the chemical just sits in the gap.
Exactly. It forces the neurotransmitter to remain active in the synaptic cleft for a much longer duration.
Why would we want that?
Well, severe depression has been consistently linked with reduced levels of serotonin. So a very common treatment is SSRIs, selective serotonin reuptake inhibitors, like Prozac or Zoloft.
Oh, SSRIs. I know those.
Right. By preventing the cleanup of serotonin, SSRIs ensure that whatever limited amount of serotonin the brain is producing has a much higher chance of bumping into the receptors on the dendrites and activating them.
Okay, to make sure this is crystal clear for you listening,
Yeah.
let me throw an analogy at you. Tell me this works.
Let's hear it.
An agonist is like a lockpick. It's not the real key, but it can open the door. An antagonist is like jamming a piece of chewing gum into the keyhole, nobody's getting in, and the door stays shut.
That's spot on so far.
And a reuptake inhibitor is like a stubborn bouncer at a nightclub who locks the front door and traps all the neurotransmitters inside the party, refusing to let them go home, so they are forced to just keep dancing and interacting.
The bouncer trapping the neurotransmitters is an absolutely brilliant way to conceptualize an SSRI.
Thank you.
Yes, biologically that is precisely how it functions. But it's vital to add a caveat here that the textbook emphasizes biology is incredibly messy.
Right.
These psychotropic drugs are not instant magical fixes.
Right. Locking the doors to the club doesn't instantly make the party a success.
Exactly. Often an individual has to take an SSRI for several weeks before the chemical environment stabilizes enough to show any improvement in depressive symptoms.
That takes a lot of patience.
It does. There can be severe side effects, and human bodies vary wildly in how they metabolize and respond to these chemicals. That is why clinical research consistently shows that these medications are most effective when paired with psychological or behavioral therapies.
Because they work together.
Right. The medication adjusts the chemical soup in the synapse, but the therapy helps structurally rewire the actual neuronal pathways over time.
So what does this all mean for you listening right now? I'm going to drop my student persona for a second and just look at the massive journey we've just taken.
We've covered a lot of ground.
We really have.
Right.
We started with the glial roadies building the scaffolding of your mind. We met your 100 billion neuron rock stars.
Yeah.
We watched a microscopic spark of sodium and potassium build tension and then jump down a myelinated axon like an unstoppable text message. We saw the electricity reach the edge of a void, release a shower of locking key chemicals, and trigger the next cell in line.
It is a microscopic relay race happening trillions of times a second in every single human being.
And the most profound part is that every single memory you just formed about this deep dive, every sudden realization you had, was literally forged by sodium rushing into cells and neurotransmitters floating across synapses in your brain over the last 15 minutes.
That's amazing.
The hardware is observing the hardware.
Which leaves us with a fascinating, almost haunting final question for the listener to take with them today.
Oh, let's hear it.
We've just mapped out the strict mechanical biology of thought. We've shown how every mood, every memory, and every physical action is fundamentally tied to the release of specific chemicals locking into specific receptors triggered by automatic, all-or-none electrical spikes.
Right.
So if everything you feel, everything you think, and every choice you make is ultimately governed by the rigid laws of physics and microscopic chemical exchanges, where exactly within all that predictable biology does your free will actually reside?
Wow. Leaving us to wrestle with the heavy philosophical mysteries of our own nervous systems. I love it. Thank you to everyone for joining us on this deep dive. Keep questioning the vast world around you, and never forget to marvel at the microscopic electrical storms happening inside you too. We will catch you next time.